SKIP-NMD is an initiative funded by the European Union whose aim is to restore dystrophin production in a subset of DMD boys by using novel exon skipping technology. In the following webinar, the leading scientists here in the UK, Professor Francesco Montoni in London and Professor Volker Straub in Newcastle, will introduce the project to you, but will also explain in detail what is going to happen during the clinical trial that is part of the study. So this webinar will not only be a great opportunity for you to learn more about this initiative, but also get a better understanding in general of how clinical trials look like and the tests that are being done to find out whether a drug or particular treatment has any benefit. I hope you enjoy it, and we will have some time to take questions at the end of the presentation. So now just a final note on the technical side. If you have any problems, please click the raise your hand button in your menu and you can talk to us separately. So now over to Francesco first and then Volker.
Good afternoon. Nice to talk to you from London. I'm here together with my colleagues, Dr. Ricotti and Dr. Tang, and I'm going to take you through the slides, or a part of the slides, and then I will share some of the slides together with Professor Volker Straub. So if we can go to the next slide, Neil. So we will explain in this webinar both general aspect of what a clinical trial is, but also more specifically what exon skipping drugs want to achieve and what we want to achieve in the particular trial called SKIP-NMD, that is a new trial funded by the European Commission and done in collaboration with Sarepta Pharmaceutic and with 4 centers, 2 in UK, London and Newcastle, and 1 in France, in Paris, and 1 in Rome. But before we go there, we need to explain the complexity of doing a clinical trial. And a clinical trial, clinical trials are studies that are used to find whether new drugs might be both safe and effective. And during clinical trials, these potential drugs, when we say potential drugs, means these need to be understood whether they will work as drugs, are given to participants, in Duchenne boys in this case, by clinicians, and we monitor very carefully whether these new drugs are tolerated well by the children. Can you, Neil, next slide please? And there are different stages of the clinical trials. There are the first stage of starting, if you like, to see whether a drug is sufficiently safe to be taken to the next stage. That is a phase 1. So this will be relatively small study that are done both to check if the drug is well tolerated, but also what is a dose that can then be given to a larger group of people. And the phase 2 trials is exactly that. Having identified an optimal dose that clearly has to be sufficiently safe, then a larger group of people is studied in a phase 2 trial where we continue to assess safety, but also we start to look at potential signs of efficacy, while the phase 3 trial is the last stage where even larger studies is done to definitely confirm the clinical efficacy. Next slide, please. So the clinical trial, therefore, are there to test the hypothesis that a new medicine may be effective and, of course, safe. We measure first how individuals are affected by a particular disease. In Duchenne muscular dystrophy, we will check if like some aspect of clinical severity of these boys, and then we measure this before and after the trial is finished. In order to try to be clear that the effect is due to the new medication, we often compare the children who receive the treatment to children who receive just a placebo. That means is not an active compound. It's just a compound that pretends to be a drug, but is not. It's just a compound that has no efficacy. And then when we start to collect information related to whether the potentially new drug is behaving better and is safe compared to the placebo. And this is how the trials assess new drugs. Next slide, please. The challenges of doing a clinical trial in a condition like Duchenne is that Duchenne is not a very common disease. Secondly, some of the intervention target only a small amount of a patient who will be eligible. So there is the need to have children recruited in many centers. In this particular case of the SKIP-NMD, there will be 4. But this means that we need to ensure that tests that are performed, say, in London, in Newcastle, and in Paris, and in Rome, are done in the same way, that we measure exactly the same thing. Also, that we harmonize the care, meaning the children should receive the same, if you like, standard of care regarding, for example, the medication that are offered, the amount of physiotherapy these children receive, so that you can compare a group of children who are quite similarly cared in different centers. In this study, like in the SkipNMD, in addition to just measure how strong people are, how long they walk for, and so on, we will also measure new biomarkers that could give us indication of clinical efficacy. And some are biomarkers that can be studied in blood, and others are biomarkers that can be obtained by looking carefully at scans of the muscle using magnetic resonance imaging. Next slide, please. I think that I would now ask my colleague, Professor Wolkestraub from Newcastle, to carry on.
Okay, hello everyone, and thanks for listening in. So you will realize that we switched from an Italian accent to a German accent. Well, most of you will, of course, be familiar with the clinical signs and symptoms of Duchenne muscular dystrophy. But just to briefly describe again what's the underlying problem, because this is really relevant for the trial we are talking about. So Duchenne muscular dystrophy is a genetic disease, and the genetic change is a change that affects a gene that we call the Duchenne muscular dystrophy gene, often also referred to as the dystrophin gene, because it this gene is responsible and informs the cell to make a substance, which we call a protein, in skeletal muscles that's called dystrophin. And this substance is important for skeletal muscle fibers and also for cardiac muscle fibers, because it protects them from damage that is often induced during contraction. So when you're missing dystrophin, then you will experience more damage, primarily in your skeletal muscle fibers, but also your cardiac muscle fibers, which over time will lead to weakness and wasting, the main symptoms of Duchenne muscular dystrophy. Next slide, please. So the concept of exon skipping has been discussed over the past few years, and it's really complex to explain in an easy way. Basically, within a gene, you have different components called exons that are relevant to inform the cell how to make the protein. So each of these little exons contributes information to the protein dystrophin. Now, in Duchenne muscular dystrophy, and that's probably animated in this slide, often some of these exons are missing. Now, when these exons are missing, what you can see is then that the information from, in this case here, exon 51, doesn't connect anymore to the information in exon 53, keeping in mind that this information is important for making the dystrophin molecule. So as you could see in this graph now, when the 51 is sliding towards 53, this doesn't connect. So what exon skipping does, and if we can animate the slide again, is we use a small molecule called an antisense oligonucleotide that basically covers the exon that we want to get out of this genetic information, so to allow that the information from 51 can connect again, and here you can see now sliding together to 54. So we are now missing information between 51 and 54, but as long as this information is still, as we say, within the reading frame, you get a functional product, and we can go to the next slide. And the functional product is a smaller form of the dystrophin protein, but it is good enough to have function. And we already know this because there are also patients who have smaller dystrophin proteins, and most of you will have heard that these patients are called or have a disease of Becker muscular dystrophy, so a milder form. So what we would do with exon skipping is to restore a milder form of dystrophin, and so ideally have a muscle that isn't like Duchenne muscular dystrophy, but like Becker muscular dystrophy. So this doesn't really represent a cure, but hopefully is able to protect muscles better than without any dystrophin. Go to the next slide. And so this project, SKIP-NMD, and Professor Montoni already mentioned that this was funded by the European Commission. So the European Commission is keen to support collaborative research across countries as well for patients with rare diseases. And Francesco Montoni applied for funding and was successful. There are several partners involved in this project, and the main objective is to develop an antisense oligonucleotide, and the name that you see there, SRP-4053, refers to the product name that Sarepta, one of the partners, is currently assigning to this compound. So this specific oligonucleotide, similar to the scheme that we just saw, would skip exon 53 of the dystrophin gene. Now, as I said, the major objective of the project is to actually conduct a clinical trial. While we are doing this, and because this is the first time this specific drug will actually also be administered to patients, we will also make sure that we develop the best measures that can look at efficacy of the drug, both by assessing the patients, but also by looking at blood to see whether there are, for example, substances and molecules that show us whether there's an effective positive response of the drug, but also to look at safety aspects, as mentioned before. Next slide, please. So I've just mentioned this is the first time that this compound is or will be administered to patients, but the reason why we are confident that there might be efficacy is because it has been tested before in patient cells, and in those patient cells it has shown that it can actually effectively skip this exon 53. We have also looked whether animals that were injected show any side effects, because safety is one of the most important aspects whenever you administer a drug the first time in a human patient. And so the first part, and Professor Montoni already mentioned these different phases of clinical trials, of the SKIP-NMD project will be a phase 1 trial, so the first time that this drug is administered and given to patients. Francesco, do you want to talk about the partners?
Okay, back to Italian accent. As you already understand, this is not the initiative of a single group, of a single university to take a new medicine forward. Is a complex task that requires collaboration, apart from the funding, but collaboration from people from different institutes. And all the symbols that you have in the bottom of the slides give an indication of the complexity of doing taking forward this initiative. On UCL and Great Ormond Street Hospital is, if you like, the two centers in London that are involved in this in UK, together with the Newcastle University, where children will also be treated. The other clinical sites will be University Cattolica on the right-hand side and Institute of Myology in Rome and Paris, respectively. The other symbols that you have are symbols of universities such as the Royal Holloway that have contributed to the design of the antisense oligonucleotide. They are symbols of companies such as Charles River involved in assessing the safety of this compound. They are CISNAV is involved in developing novel outcome measure, novel way to measure how people move and how far they move, so that we can obtain a very precise indication of this. The CRIS, Chris is involved in imaging technology, and Sarepta is the drug company that is producing the novel antisense oligonucleotide to skip exon 53. Next slide, please. And many of you will know that there are different compounds that are being developed to induce exon skipping. And some of you will be aware that for exon 51, that is a different exons that can be also targeted by antisense oligonucleotide, two different initiatives have been or are ongoing. One that is the one on the top, the GSK ProSensor compound, and below the Sarepta compound. We thought it was important to highlight that even for exon 51, there is no definitive evidence that the antisense are effective for the GSK ProSensor antisense. Early studies were quite, if like, favorable. Phase 2 studies were quite favorable, but a phase 3 study appeared to be less favorable. So the question is, why is that? And the slide mentions that further analysis of a subset of patients is ongoing. And ProSensor recently provided some explanation that are, I think, plausible explanation of why the patient recruited in the phase 3 study that were probably a little too advanced, and within the year or the 48 weeks of the clinical trial, it was really not possible to detect a improvement, while observation for longer in a subset of this patient indicated that most likely is clinical efficacy. The exon 51 study below that one done by Sarepta is done with a molecule, with a chemistry, as in one of the next slides will be explained, that is slightly different and is a chemistry that use a different backbone. I think if you just go back, please. I think yeah, just go back again. And again. Okay, so the exon 51 from Sarepta was a smaller study. The result appeared to be very encouraging, although the number of patients studied was smaller compared to the GSK Prosensa study. What was clear is that children treated with this exon 51 drug developed by Sarepta appear to have probably more dystrophin than children studied in the previous study. But again, these are still relatively small studies. So I think one has to take into consideration that a definitive answer of the clinical efficacy of this compound is still pending. Next slide, please. I mentioned before that there are some differences between different compounds developed by different companies. Even if both Prosensa and Sarepta have developed a drug for exon 51 and now are developing a drug for exon 53, there are subtle differences in the chemistry with which these antisense are actually manufactured. So some of these antisense differences help to maintain, if you like, the levels of the antisense in muscle for longer, and may be associated with a different profile of side effect. And the specific chemistry that Sarepta has developed is called morpholino. And this is the chemistry we will use for the SKIP-NMD trial. Next slide, please. So the exon that we will target in the SKIP-NMD is now going to be exon 53. The specific chemistry is going to be very similar to that Sarepta used for the exon 51 clinical trial. We will give both the active compound and the placebo drug in the phase, in the first 16 weeks, where we are trying to find the optimal dose for this drug, but also assess its safety. So that will be a part of the study that we call blinded. And after this initial phase that will last a few months, all children will receive the active compound for another 48 weeks. Also the children who had been originally given the placebo. The drug will be given weekly by intravenous infusion. We will have to measure the difference that these drugs makes in the production of dystrophin. So we will need to compare a biopsy before and a biopsy after the study. But also we will look at scans of the muscle to see whether there is any improvement, if you like, that we can detect on muscle MRI in this patient. Next slide. As Volker mentioned, this drug has not been given before to people. So one of the partners that I mentioned earlier on made a number of studies in cells and in animals to make sure that all what can be anticipated of a potential toxicity is being studied carefully. And of course, there will be no study if toxicity or significant toxicity is predicted. In addition to some of the physical activity measurement that many parents and children will be very familiar with, during the clinical trials, we will also measure things that we hope will allow us to detect the response to treatment, or any response to treatment, a little more precisely than what we do currently. So we are, as the slide says, we are testing the test because we anticipate that some of these tests may work better than just the ordinary clinical tests that we do in clinic, but in reality we do not know. So that is why the study still is testing new tests. And I will ask now Professor Straub to continue with the next few slides.
Okay, so the next slide is basically a very brief summary of what we have already said. So SKIP-NMD is a first in man, as we say, a first in patient study, therefore a phase 1 clinical trial to primarily look at how safe the drug is, but of course all of us are also interested to see whether there's any efficacy and effectiveness of this drug. And this drug is an antisense oligonucleotide, often referred to as a molecular patch because it covers this exon 53 so that it's not part of the information of the dystrophin protein. And it was developed by Sarepta, and Professor Montoni already explained that there are different chemistries that define those substances. So the next slide is then, well, who can actually participate in this clinical trial? The centers that are involved are across 3 countries, the UK, Italy, and France. The idea is to recruit 24 boys. And now, for many families, it's always important to understand, well, who are those boys who can participate? We don't really have the luxury even to pick from a large cohort of boys, because for this drug you of course need to have this specific genetic change. There will also be other inclusion criteria, and we'll come to them in more detail at the next slide, but it's already summarized here, that make sure that the boys that get the drug are actually comparable to each other. The age that we have picked should be between 6 and 15 years of age. But you also need to be able to walk at least 250 meters in 6 minutes. So just because you might have the right age doesn't mean you are necessarily included. You need to, in addition, be able to walk 250 meters. It's also important that you don't have any significant problems with your heart or with breathing. And then we will look at these boys that are going to be treated compared to boys that are untreated. So coming to the next slide, some of these inclusion criteria are listed in more detail. On the next slide, please. So we already mentioned that it's a prerequisite that you have to have the correct genetic change that is skippable for exon 53. You need to be aged between 6 and 5 years, walk for at least 250 meters, shouldn't have any respiratory or heart problems. And then for the study that doesn't include boys who are treated but just are compared for the natural history, you basically have similar inclusion criteria except for the fact that your genetic change doesn't need to be amenable to exon 53 skipping. If we can come to the next slide, please. That lists where the centers are. I mean, we have already mentioned at the beginning that London and Newcastle are the centers in the UK. So for those of you who are calling in with boys who would have the genetic change, these are the 2 sites that you could pick from. Then there are centers, as we said, in France, in Paris, and in Rome, in Italy as well. It's important that no matter whether you are far away from any of these 2 centers, that any expenses that would occur during traveling, and if you would need to stay overnight as well for accommodation, would be covered by the trial. Next slide. So how could one make sure that you are picked for the trial? Well, it's not just a question of being picked. It's a question of really being tested whether you would meet all the inclusion criteria or your son. So therefore, before the trial, it's important to basically understand everything that we are discussing in the webinar, and all of this is summarized in a patient information leaflet, also a leaflet for parents. And if you fully understand what the trial means, then you're asked to sign a so-called informed consent form, and then your son will be assessed for these inclusion criteria. Now, it's also important that the boys themselves do actually have an understanding about the journey of a clinical trial. And the ethics committees want us clinicians to specifically also get assent from the boys. So often parents are asked to and expected to discuss a clinical trial beforehand with their children already and with their boys, so that the boys are prepared for this. But throughout the trial, there will always be an opportunity to ask more questions. Next slide, please. So often you might already know the center in London or Newcastle. If not, then the first visit will serve to inform you about the trial, sign the informed consent form, and inform you about your schedule. So the trial will last for 48 weeks, so it's quite a big commitment for a family and for a boy as well. And for this 48-week trial, you will get a almost clear agenda that will inform you when the boy would receive the drug and what kind of assessments will be done. It doesn't mean that all the assessments will always be done at each visit, but the schedule will specifically list that at some visits there will be more investigations than at others. So every 4 weeks, as we list here, participants will be asked to complete a series of tests, and they will all be monitored by healthcare professionals, physiotherapists, and the clinicians. And after the 48 weeks, and this is of course what everyone is interested in, the data will be analyzed. Now, this also means, of course, that all the participants need to have done the 48 weeks. So if you are a patient and you have already finished 48 weeks of the trial, it doesn't mean that you will automatically get the data. We will look at the data from all the participants and then make sure that everyone is informed about the outcome of the trial. Next slide, please. So we will come to some of the tests in a bit more detail. In the entire discussion about antisense oligonucleotides, the 6-minute walk test had already a lot of attention, because the regulatory authorities that finally approve that a drug can be sold on the market and can be accessible for patients, they have said that walking is a very meaningful readout for testing the efficacy of a drug. So I don't know whether the video works. Basically, what a boy is asked to walk as long as possible, and it's not about running but walking, for 6 minutes, a defined, and this defined distance is marked by the two cones that you might see in the front and at the end of the corridor, and there will be a physiotherapist going to also make sure that the boy doesn't fall. And so it's quite a boring exercise, but we know that it is fairly reliable as well. So this will be done, the 6-minute walking test, at several time points throughout the clinical trial. The next slide, please. Another test that will be done is a so-called magnetic resonance imaging, MRI scan. So here we look at the muscle by an imaging technique that allows you to basically look at slices throughout the legs of the boys. We hope that this will give us important information about the efficacy of the drug. Now, what you do, you go onto into the machine, or this, yes, and it looks like a donut shape where you are basically sliding in. The only challenges are here that it's quite noisy, and if you are afraid of narrow spaces, but we know that the boys actually tolerate this quite well, and you can hear your own music as well. So this scan will be done a few times throughout the course of the trial as well, 4 times, 3 times altogether. Yes. Next slide, please. And then we will also do muscle biopsies and skin biopsies. Now, often the muscle and the skin biopsy are taken at the same time, so it's not an additional burden really. But of course, you need to be informed about, and the parents and the boys, that tissue is taken. And the biopsies are so important because they will show whether the drug was able to get dystrophin reproduced in the muscle. So what you see here in the bottom lower corner, in the right lower corner, is that dystrophin normally looks like a nice white signal around the muscle fibers. The cross-section of these muscle fibers look like circles, and dystrophin is very strongly visible there. You can also see that a patient with Becker muscular dystrophy has less dystrophin, but a lot more than the boy with Duchenne muscular dystrophy, DMD, where there's no dystrophin at all. Sometimes we also take biopsies from carrier moms and can see that some of the muscle fibers don't show any dystrophin, and this is a good example. Many of the carrier moms don't necessarily have any symptoms, so you don't need completely normal dystrophin to have fairly good muscle function. The next slide. And then, of course, many of the tests that we do are based on really blood sampling and looking at substances in the blood that will tell us whether there is either less damage in the muscle fibers, or we are also looking for molecules or substances that might tell us that the muscle fibers are better sealed. So part of this is also done to look at the safety aspects. So when blood tests are taken, to make sure that we monitor how, for example, the kidney and the liver work. And the next slide.
So now we're going to do a blood test. And we've had the anti-hoc, that's the one we like. And this is the endocrine, just want to show you that takes a little bit longer. For some people that have bit sensitivity to that. And then this is the spray. So if you didn't want to use cream, or you came in with that cream on and you want to test it straight away, then this is the magic spray that just makes it very cold. Okay? You don't feel it. But if you have the cream on, let's just do a test. Okay? So this is where it's in here. pump in the vein, then the blood can come straight out. Okay? Once she does the blood test, we can also have a look at the book. Okay? And try and find Wally, and I have here as well. Okay?
So I mean, you could see here how the nurses try to make sure that the boys get everything explained. Now, in the next slide here, you will see that we specifically also look at muscle function, for example, in the upper arm, and there are two small videos that are animated that we will play now. Well, if there's no specific audio, basically in these tests, again, boys are instructed on what they should do, and these tests are done throughout the course of the trial several times, and so your performance is always compared to your baseline performance before you were treated, and then to see whether there's any efficacy of the drug. And then if we go to the next slide, yes, so we will also keep a close eye on the heart. I already mentioned at the beginning that dystrophin is present in the heart, and you will know that in Duchenne muscular dystrophy the heart can be affected. So we want to also make sure that we closely monitor the heart function to see whether we can see any effect of the drug, and also to make sure that there is actually that it's safe and that there are no problems with the heart. So these were most of the tests, and I will hand over to Francesco again to discuss, you know, if you change your mind during the course of the trial.
Yes, thank you, Volker. Next slide. So this is true for all clinical trials, and the same therefore applies to SKIP-NMD. Participation to the clinical trial is voluntary, and it's not a mortgage that is written for the next 25 years. People sometimes change their mind. They feel that either the study is too heavy in terms of the frequency of the assessment, or for other reasons. Sometimes family circumstances may change. And it is important that the families and the children are aware that to withdraw from a clinical trial is completely normal, and they can talk to the study nurse, to the physicians, to the parents, and let us know, and this will not be will not affect the way that children are and families are cared for in the center where they are seen. Next slide, please. So in terms of a question that comes very often is, would this drug be available to the people who have taken the drug during the study, but also to other individuals once the study is finished? The difficulty is to be completely, to say that this will be definitely what will happen. The reason for this is that if the drug was somehow not very effective, for example, there will be very little perspective of this drug to be produced for the next 20 years or whatever, because there will not be the reason to produce this drug. So that will be, in the case the study showed that the drug was not very effective, I think it is not likely that the drug will be available for the children who have been recruited in the 48 weeks of the study, but also for other children. At the same time, it is in the interest and the intention of the drug company to ensure that the drug, if it works, will eventually get into a path for authorization. And the study the SKIP-NMD study is not per se a study that will automatically lead to authorization of the study drug. So it will be in the interest of us investigators and of the company to increase the number of children studied, and most importantly, increase the number of months that children have been treated for, because 48 weeks is a relatively long period of time, but the longer the treatment is, the more the divergence between the natural history of the condition and the treatment effect should be. So it will be, the intention therefore is to continue treatment if the drug is effective behind the 48 weeks. But in all honesty, this cannot be absolutely guaranteed now. Next slide. Another point that is often asked by families is how will they know how the study went. I think that there are a number of different ways to do that. Firstly, information related to the study is communicated to families when they come in the research facility at the end of the study, when the results are available to us investigators. Also, the result, there is the interest, in actual fact is a requirement that the studies are communicated to the scientific community. And this is both if the results are positive and if the results are negative. This is an important part, because it is important that the community learns both from successes but also from problems in case there have been problems. Clearly, the information regarding what is presented at scientific meeting and in publications will not be identifiable data, so it's not possible to tell this is my son, because the data are anonymized. We clearly work very closely with the parent organization, and the result will be shared with the various parent organizations with which SkipNMD works, and therefore that will also be another way to disseminate the result. Next slide, please. So before we go to any questions, I just remember that I've forgotten to, at the very beginning, to mention the fact that the effort of the SkipNMD are not only efforts of scientists and doctors like us and the colleagues in Newcastle and the other colleagues that I have thanked, but also we have worked very closely with the various parent organizations, and the parent organizations have helped us to identify potential ethical problems and so on. So they work a little as colleagues to point out to us investigators areas of potential conflict, of areas of potential problems that require our attention. So firstly, I would like to thank the Muscular Dystrophy Campaign also for hosting this webinar. In UK, we also, and Muscular Dystrophy Campaign has worked very close with Action Duchenne and the Duchenne Parents Support Group. In France, we have the AFM and the Duchenne Parent Project that also is active, very active in Italy. So as an organization, as a principal investigator of this study, I think I should thank them in particular. And then just before concluding this, I also would like to thank a lot Cathy Turner from Newcastle, who really helped a lot to shape and focus our mind on information to disseminate to families. So I have nothing else to say, so I will now pass on to Neil and Marita at the Muscular Dystrophy Campaign headquarters.
Okay, thank you very much, Francesco and Volker, for this very clear and fantastic presentation. And I have to say as well, I mean, from the Muscular Dystrophy Campaign, we are very kind of proud and honoured to be part of it, to represent together with the other organisations the patient voice. So I think now we have time for some questions. I just kind of hand over to Neil for a couple of minutes to explain how you can put questions forward, if you like.
So there are two ways you can ask a question if you're watching online. Firstly, you can type it into the chat box. It'll appear here to us and we'll be able to read it out to Francesca and Volker. Alternatively, if you click the raise your hand icon, we'll be able to see that and we'll be able to unmute your line and you'll be able to ask the question yourself. So I think Marita has one question to start with.
Yes, maybe to Francesca or Volker. I mean, can you kind of maybe comment a bit on the timeline? Where are you at the moment in the project, and when is the clinical trial actually to happen? And could you comment on this, please?
Yes, the clinical trial will start in UK before the other countries. The documentation regarding the safety aspect and so on has been handed over to the various regulatory authorities and to the ethics committee. The ethics committee, as of August, has favorably reviewed the documentation, and now there are in reality probably another month and a half of other aspects related to activating the study in London and Newcastle. So realistically, we could expect to start dosing children in the dose escalation study in London and Newcastle before the end of this year. We expect that by then Rome and Paris would have had their favorable evaluation from their committees, their national committees and so on, and therefore they will start dosing, possibly also before the end of the year, but certainly just shortly after we start in London and Newcastle.
I think we haven't received any questions so far, but Neil has another question.
So, do you think exon skipping will be expanded or extended into other exons, Francesca or Volker?
So the question is whether the antisense oligonucleotide will be, apart from exon 51 and exon 53, whether there would be expansion to other exons. I think here, again, it's important to realize there are efforts both from Sarepta, that is the company we are working with for the SKIP-NMD, and Sarepta is very close to start, most likely in the States to start with, a clinical trial for exon 45, and they are also a little behind in terms of the timeline, but will most likely start in 2015 for exon 50. The other companies involved, Prosensa, has already an active exon 44, 45, and 53 clinical trials. These are at the moment at the early stages of, if you like, assessing safety and dose finding. So there are certainly more exons that are being targeted than just 51 and 53.
I have maybe one question which we always hear, this is Marita again by the way, which we always hear from our parents. So you mentioned, Francesca, I think that if this trial shows any benefit for exon 53, it's very likely that, that the boys who are involved in the trial will have access or will continue to receive the drug. But what, if it is successful, what are the next steps? I mean, do we have to wait then another kind of 5 years and do like a phase 2, phase 3 trial with this Exon in order to bring it to the for the drug to be brought to the market? What's your expectation of, if successful, making this drug available to other boys and also men with Duchenne muscular dystrophy?
Well, this again is a important but also complicated questions, because the approval, drug approval for this group of medication, the antisense oligonucleotide, is a moving target. Moving target not because the regulator are being difficult, but because new evidence is accumulating as every few months. So I think that the recent deliberation from the FDA, that is the Federal Drug Administration, regarding Exon 51 drugs, so this is not 53, the SKIP-NMD, but Exon 51, have, if you like, open a window for accelerated approval of Exon 51 drugs, but have also indicated that they would like to see a separate randomized placebo-controlled study, meaning that even if the result of the SKIP-NMD, for example, were interesting and evocative of efficacy, that might not be sufficient for the drug to get into the market at that point, but a larger study might be required. At the same time, and that I think is important, because it may well be that will be the scenario we will be in, say, early 2016, when the SKIP-NMD study will be finished. Having said that, there may well be additional evidence that for the Exon 51, but also other studies that have been done. Again, as I mentioned, there is a parallel study for Exon 44, 45, and 53 from ProSensa, and there is an Exon 45 study that will be started very soon by Sarepta in the States. So there will be a set of different evidence at that point in time that may change the way regulatory authorities will see the approval path for this drug. So, in conclusion, in a conservative way of looking at it is to say, well, a possibility is that after our study is concluded, hopefully with positive result, a larger study may still be required, and that will be another study that will realistically take at least a year and a half, probably a little longer, to demonstrate definitive efficacy. However, it's not impossible that by early 2016, the level of evidence that will be available to us and to the regulator about the efficacy of the antisense oligonucleotide may be different compared to what we have today, and that the path to approval may be not as long as in the first scenario I have highlighted. However, in reality, we do not know. So it's important to start the journey being aware that it may be that it will take years even after the SKIP-NMD is finished in order for the drug to be more widely available to the population of people with Duchenne.
Thank you very much, Francesco, for that very, very kind of comprehensive answer. So I think you must have given a really, really good, easy to understand presentation, because we have still not received any kind of any questions. So obviously there are no kind of unclarities there. I mean, can we maybe hope within SkipNMD? Do you think like this update will become now an annual update? So can we expect maybe next year around this time to have another webinar? Would that be realistic?
Well, Newcastle. I mean, the suggestion to make an experience that, for example, clinical trial and the colleagues in Italy had in the past, where if like the interest in having information disseminated via the webinar that has the advantage that can be accessed again without needing to be available online now is. So in a way, we will be very, you have been very kind to organize this. We've certainly been willing to share the information we have. The colleagues in Newcastle have done a lot of work to facilitate the putting together these slides. And if there was the desire, we would certainly be able to do additional webinar in the course of the study. I think an important point at the same time is that it is actually not legal to publicize or to disseminate information regarding the ongoing study before the study is closed. So I think that it is possible to give information en route, if you like, on how many children have been recruited, whether the drug has been well tolerated, and so on and so forth. However, it's not going to be possible to give indication whether the drug is, in inverted comma, working or not working before, because this is not feasible. So I think that that may be an important point. Maybe an issue that could be rediscussed in a few months' time is that if families have some specific questions and so on regarding what are specific inclusion criteria, the studies and so on, we could certainly host other webinars, or we could respond to specific queries of things that maybe we haven't covered in this webinar in future webinars. So perhaps I'll suggest to ask the colleagues in Newcastle to comment further.
Yeah, I mean, just to add that the SkipNMD project is more than just the trial, and we will also provide news updates through the SkipNMD website. So there's a website that can be opened by anyone. It's a public website, and so we will continue to update the public in late term news updates about the progress of the project overall.
Thank you very much, Volker. Yeah. So just kind of for clarification, there is the SkipNMD website. If you Google SkipNMD, and that will be updated. But also, kind of, if you do kind of feel like that, thinking about what you've heard, you might come up with one or the other question. You can always email our research inbox, which is research@muscular-dystrophy.org, and we will then forward the questions to Volker and Francesco, and I'm sure they will be happy to answer. So I think, yes, so just my closing remarks, I think this is a very exciting study. It's fantastic to see all the scientists working together internationally, and also kind of, I'm very pleased, as I said before, that the patients' organizations have been involved from the start, and from our point of view, this has been a fantastic collaboration, and so I would like to see this going on like this. And so many, many thanks to Francesco and Volker again for all this hard work, but also kind of to Kathy Turner in Newcastle, who has been fantastically in putting putting this together, and also Valeria here in London, and Neil Bennett here at the Muscular Dystrophy Campaign. So of course kind of a presentation like this is more than a couple of people that are involved. And I hope we will give you another update, as Francesco and Volker said, on at least any progress, hopefully in the next year at some point. And until then, I would like to thank you very much for your time and your attention. And if anything if you have any questions, just let us know.